Good morning, everyone. My name is Reza Owisi. I am assistant professor in the Department of Food Science and Technology at Texas A&M University, and today I will be talking about cellular agriculture, which is an emerging technology in food biomanufacturing, and it is revolutionizing the food production systems with different types of applications. I will go through that and I will try to explain how it works. So before jumping into the cellular agriculture, I need to actually bring some of the challenges that we are facing in food production systems. The world population will be 10 billion by 2050. We have problems with the water production, emerging diseases, the climate change, how much water and resources we are putting into the systems, and how much food material we are harvesting, the waste stream, emerging pathogenic bacteria, and also the problem with the fresh water, the available fresh water. So all of putting these together always actually motivated me to focus more on developing sustainable food production systems and finding a solution to address some of these challenges. By 2050, in order to be able to feed 10 billion people, we have to increase our food production by 70%, and our meat production must be double technically. It must be increased by 100%. And at the same time, we have to reduce our meat consumption by 70%. But the problem is the consumers. So technically, many people they don't want to give up meat, and that's the challenge that we have. So in this case, we have to find other solutions to address these needs and provide some other technologies that we can create meat but not in a conventional way. So again, that's another slide that we put together specifically for the seafood. The seafood challenges is a little bit different from the meat challenges. For the seafood, we have problems with the microplastics, the waste stream, illegal activities, permits. For example, in the United States, more than 90% of our seafood is imported from other countries, and we don't have that much control on the chemicals that they have been using. It's very difficult to control everything. So in order to be able to produce more safe, sustainable, and clean meat and seafood products, and even other agricultural products, we can really actually use this technology. So, but there are different types of food biomanufacturing and methods of the food production that they are emerging at this step. For example, fermentation is one of those methods. We are using fermentation to produce fats or oil or protein for different applications. Insects protein. So another program that I have in my program, we are another topic that we are focusing on in my program is just focusing on the insects protein and insects processing. Vertical farming, and that's very unique and very neat technologies for food production, plant-based protein, but emerging technologies like cellular agriculture can also be actually categorized under the emerging technologies. But what is really cellular agriculture? Cellular agriculture, as it sounds, technically depends on the cells. We take the cells from the animals or from the plant tissue and we bring them to the lab. We reprogram them because the moment that you take the cells from the animal, for example, muscle cells or fat cells, and bring them to the lab, they are not muscle and fat anymore. We call them de-differentiated. So they already been differentiated to the fat and muscle when they are in the inside of the body of the animal, but once we bring them into the lab, and so technically they de-differentiate, and we don't have the same fat and muscle. So technically we have to keep them for a long time, and we have to work on them and make sure that they don't die anymore, which is called immortalization, and I will go through that. And then after that, we transfer them to media and bioreactors. We produce them in the mass biomass and then put them together in a scaffold, and then after that, so we can have different types of products. But going through the history of the cellular agriculture, the first person who actually mentioned cellular agriculture was Winston Churchill, and he mentioned that instead of just producing the whole chicken and just separating the chicken breast, and the rest of that is going to be just wasted. So what if we just grow the chicken breast? So we can find the technology to develop that. Then little by little this idea was developed, and in 1998 one of the researchers at NASA. So he published a paper eventually in 2001, and he talked about the cellular agriculture as one of the best methods of the food production for a space mission and colonizing the Mars. But in 2013, Mark Post, who is also considered as the father of the cellular agriculture, tasted the first cell-based burger on TV show in Netherlands and in front of millions of people. And also in 2015, he had a talk at IFT, and I was there in his talk, and probably thousands of people actually they were there as well, and he introduced this topic to people. So the first burger actually cost $325,000 to make. So it was made by Mark Post. He's one of the leaders in this area, and I'm really honored to have a publication with Mark Post as well. So our group also is one of those few groups that started focusing on cellular agriculture at early stage. So when we talk about the cultivated meat, so technically speaking, the greenhouse gas emission is much lower compared to the conventional meat production systems. The water application is much lower, and also we don't need that much land. 99% less land is required to produce cultivated meats. When we talk about the cellular agriculture, sometimes people they just think actually this is just related to the meat, but it's not true. So we can actually have cell-based coffee. There is a company right now that they are producing cell-based coffee in Seattle. Cell-based chocolate, as you know, coffee and chocolate they are not sustainable. The current production system, cell-based milk and cell-based meat, seafood, and insects. These are all the products that we are talking about. When we talk about the cellular agriculture, it's technically an umbrella, and right now we have cell-based leather. There is a company in Portugal. When I was in Portugal for a conference, I met with them, and they are producing cell-based leather from the cells from the alligators. So technically, without killing alligators for their skins, we can have leathers that they have cells from the alligators, but they didn't kill any animal for producing that. Well, so cultivated meat technology isn't quite interesting and new. However, this technology has been around, right? So we have been using regenerative medicine technologies for regenerative medicine for a long time, for decades, and they are very, very advanced. I can give you an example. In 2018, I approached a professor in regenerative medicine, and I tried to convince her to work with me to develop a serum-free media for cellular agriculture. And she told me it's already been done, so why we have to do that? Because there are lots of products in the market, serum-free media, that people they can use them. However, the problem is actually, so they are very expensive, and they have specific applications for medicine or research, but not for food production, right? So for regenerative medicine, there are lots of priorities, and when we look at the priorities that we have for regenerative medicine, it's totally different from the priorities that we have for the food production. When we are trying to translate the technology from the regenerative medicine for the cultivated meat production, we have to consider four different actually aspects: the cost and the scale up, the texture, flavor, nutritional value, and being food grade and safe. Many products that we use for regenerative medicine, for example, for inducing the differentiation, so they are not food grade. We are not allowed to use them for food production. These are the challenges that we are facing when we are trying to translate the technology from regenerative medicine to cultivated meat. So in 2021, we received a large grant, and we established the first National Institute for Cellular Agriculture. This was the largest and the first federally funded project in the world on cellular agriculture, and still it's an ongoing project. It is evolving and growing rapidly. So in my program, so we are focusing on cell line development. We are trying to develop cost-effective serum-free media. We are trying to develop biocompatible scaffold to put the cells on them and develop food safety aspects of the cellular agriculture. I'm going to go through some of these aspects and talk a little bit more about them. For example, for the cell line development, it's a very challenging topic. When we started focusing on this topic, there was no specific cell line available for cellular agriculture. Many people, they started focusing on seafood and other animals, which was very difficult, and then they were able to develop that. In my program, we already developed multiple cell lines from animals and from fat and muscle. But it's been very challenging. It's not very easy task. You have to go harvest the cells, bring them to the lab, and then adapt them to the new condition. And when we are talking about the culture conditions, we are talking about hundreds of conditions, right? And we don't know exactly what is happening. We have to actually try different methods to make sure that the cells, they survive, and then we have to characterize them to ensure that the cells characteristics and the gene characteristics don't change by passaging them by the time. So these are some of the cell lines that we have been focusing on, mainly for the seafood. One of the challenges for the seafood is the contamination. Getting rid of this contamination is not very easy. It's very difficult, particularly for the oyster. We tried oyster at least 2 times a year. I think by this time, actually, we tried this 8 times. So we brought oysters 8 times to our lab and tried to develop the cells. Contamination is always there, and it's very difficult to get rid of them. That was one of the failure that we had actually multiple times. But we have been working with different other cell lines as well, and we were quite successful so far. For example, just giving you an example, this is the samples that we collected from the lamb and right after slaughtering, and lamb fat and lamb muscle, both of them developed in my program. We have primary cell line and immortalized cell line that they don't die anymore, and you can keep passaging them for a long time. There is a difference between primary cell line and the immortalized cell line. That's immortalized cell line, they don't die at all. Maybe after passage 150 they actually stop growing, but I haven't seen that so far. And then we grow them. So we sorted our cells. So when you harvest the cells from the animals, this is not, for example, just 100% fat. You are harvesting the fibroblasts and also some other cells as well, right? So you need to actually sort them to make sure that the final product that you have is 100% fat cells. And in order to do that, we use antibodies, right, as a marker. And then we pass them through a specific equipment that we have that at Texas A&M, not in my lab. There's a core facility. And then we sort them, we sort the cells. It's called sorted cell line. So now we understand that we have 2 types of cells, unsorted and sorted, right? So many people, they don't do sorting, but in my program we do both of them. We have the sorted cell line and unsorted cell line as well. So, and then after that, we immortalize the cell line. In order to immortalize them, there are 2 methods of immortalization. One of them is just spontaneous immortalization. It means that you keep passaging the cells, right? And at certain point, they change their characteristics, and then they start changing their morphology and they grow, and then they become immortalized. Many of them, they die during this process, but those that they stay alive, they will be immortalized. However, that doesn't happen all the time. For example, for our lamb fat and lamb muscle, it didn't happen spontaneously. What we did was, at the same time, we also actually used the viruses and transfer some genes to the cells, and then we immortalize them through the genetic modification. So now we have immortalized cell lines, but probably some people, they will say, well, so these are immortalized cell lines through the genetics modification, which is true. And there is a discussion and arguments between those 2 groups, that they are pro or against GMO. But technically speaking, right now, so the best method for immortalization, the safe, rapid, and a lot cheaper actually, is gene transfer. So, and technically speaking, right now we have immortalized cell line in our program from the lab. Then after that, you need to do the PCR to see whether the genes, they are there or not, and then there are different markers for the fat and muscle that we confirm that through the qPCR as well, and we are in the middle of the transcriptomics as well. And here is this. So technically speaking, we had the scaffold from the plant-based material, and that's the best option. We call it hybrid product, which means that you have the plant-based material or fungi-based material, and at the same time you have these cells and you put them together. And we also apply different types of TVP from the pea and soy, different types of methods of the attachment, and different surface properties enhancement, for example, through the plasma. We wanted to see if we use the plasma for, can we enhance the surface properties, and it didn't work very well actually for us because it's too hydrophobic, and because of that the cells, they don't like that at all. So the regular method or regular scaffold, actually they work better. And technically we produce lamb shawarma in our program right now. We haven't tasted them yet, but we are working on the GC mask to collect the flavor and compare that to the flavor of the conventional lamb. Cell line development has lots of challenges. When you talk to people that they are working in cell line, there's an always discussion among them. So first of all, we don't have access to all of the target organisms that we are looking for. For example, I've been looking for octopus. It's been very difficult to have access to that. For example, particular for me that I am here, and I cannot travel to Florida that they have some, I have some friends over there, and go there and take some samples. I need to have that live animal in my program to be able to take the sample, immediately put them into the media, right? Having access to that is very difficult, and in many cases they are seasonal, so you don't have access to them through the year. Different species have different cell properties. Maybe lamb, human, and bovine, they kind of similar to each other, but it's very difficult to work with seafood, marine species versus freshwater species, right? Warm water species versus cold water species. These are very different and they have different physiology. It's very difficult to apply the same formulation and methods for the cell line development for every single organism that we have. Contamination, it is another problem that we always have, particularly when we are working with seafood products. Cell properties, doubling time might not be really good. Doubling time is the time that you require, that the population require to have to double their population, right? The biomass will be doubled. For example, for the lamb, it's around, in my program, it's around 17 to 20 hours. It means that after 20 hours, the population will be doubled. But for the fish cell lines, it's really long, between 40 hours to 80 hours in many cases. Again, sorted versus unsorted, I have both of them in my program, but so many actually of my collaborators, they don't want to do sorting. Some of them, they want to do sorting. So there is no right or wrong here. An immortalization process. So spontaneous versus using the gene transfer. So this is another actually topic that we need to discuss, that for example in Europe, GMO, it's very difficult to sell the GMO products in Europe, right? But in the United States, so there is no regulation against that. These are the things that we need to actually consider when we are talking about, okay, so I have this product, and if I want to actually export that to Europe, so I need to meet their requirements as well, right? This is very interesting topic. The other topic which we have been working on is related to the scaffold development, and there are different methods of the scaffold development. In my program, we took banana leaf, for example, and then we remove all of the cells. This process is called decellularization, and whatever left is just kind of the scaffold, the skeleton of the plant material, and then we added our cells to that, and then we cooked that material in our program, and we noticed that technically plant-based material, they can provide really good heat resistant product, that technically the quality of the final product won't change after cooking compared to the actual fish. We are also working with the hydrogels. We just submitted this paper. We applied different types of hydrogels alone or in combination with different types of proteins, and we measured the mechanical properties, the characterization, and then after that we put the cells on them, and so the cells, they attach very well to some of them. We have challenges with this scaffold, so the proper texture. So no matter what we do, at the end of the day, we have to provide the texture that the consumers, they like. right? This is very important. And in order to do that, we have to apply different technologies. I don't think that in the next 25 years, we will be able to produce cell-based, 100% cell-based product. So all of the products that we are going to see in the market, they are going to be hybrid, plant and cells, or fungi and cells, or a combination of all of them. Hybrid products, that's the best actually method to go with. Storage and shelf life should be studied, and there is not that much information about the storage and the shelf life of the final products of the cell-based meat. Many of these materials that we are working with, they have poor cell adhesion. We have to find a way to enhance the cell adhesion to them. It could be diffusion, it could be some materials that they can actually enhance the surface properties and encourage the cells to attach. Many cross-linking agents that we can apply, but the problem with them is there is not that many approved cross-linking agents with proper um, uh, with proper quality. Cell proliferation and differentiation on a scaffold. What we have learned, for example, soy protein can delay the cell differentiation gene expression. Through qPCR, we actually noticed that, which was quite interesting. Scaffold size and nutrient, the gas diffusion rate, these are really important topics. Lack of available studies on cooking properties. We have to work with chefs to understand what is happening with the final product and they like that or not. Sometimes we have off flavors and bitterness in the final products that we need to also address those as well. The other activities that we do in our program is media development. For the media, as you know, media can actually cost more than 95% of the total final cost, and more than 99% of the cost of the media is related to the serum that is coming from the calf, that technically we don't want to kill animal to produce meat. We don't want to use serum, and there are many other reasons that we have to stop using fetal bovine serum. And in order to replace that, we have to apply other materials. In my program, we have been working with different plant-based material, algae, yeast, mushroom, and even marine organisms, insects, and so we process them intact protein or hydrolyzed protein or fermented protein, and we use them. So one interesting topic that we focused on, we published the paper, and then after that we were able to expand it and receive another grant around that, was we, inspired by the nature, we took the samples, the gut microbial community from blue catfish, because blue catfish in the state of Virginia, they are invasive species and they eat, they feed on insects. They can digest insects with exoskeleton. We decided to collect the gut microbial community, establish that, and then ferment the insect with them. The final product is called postbiotics after fermentation, and we noticed that if we use the postbiotics, they have other components that they can promote the cell growth in the cell culture media, right? So we pre-digested that through the gut microbial community, then we fed them to the animal. Then we calculated the cost of the production, the LCA life cycle assessments. Black soldier fly, actually among all the insects that we applied, was the best insects in terms of providing the most nutrients for the cells. The cells really like that, and also it's the cheapest one and the most inexpensive. So we also applied artificial intelligence for the first time for media optimization for cellular agriculture. This paper also published in 2023, and this is an interesting topic. We are still working on that right now, expanding that for other actually applications. One important thing, if we run the experiments through the one factor at a time, as you can see here, we don't see the interaction. But through the modeling and AI, we can easily actually go beyond that, and it can tell us the interaction and the area in the space of the research that we have never actually tried that, right? This is very important. And so we tried different modeling, and we find out that out of 9 models that we applied, they were really nice and providing very good and robust models for predicting the serum-free media formulations, and it did work very well for us. We also actually applied different types of plant-based protein for bovine cells, and among them, two proteins they did work very well, and we were able to replace serum without reducing the cell growth successfully by replacing the serum by adding the plant-based protein. So media development challenges, serum-free media, that's the most actually difficult part of the work. We have to find different types of methods. That's why artificial intelligence might be actually helpful here. Proteomics and can actually also help. Different species, they have different requirements. For example, cats, they don't eat the same diet that dogs eat, right? So, or rainbow trout diets in aquaculture is totally different from the carp, right? So these are not the same. So they have different physiology. So we have to consider them when we design the media. Lack of systematic omics approach. So there's not that many actually approaching this area. A scale up is very difficult. Most of the research that people they do in the lab is around 2D area. So we have to actually translate it to 3D and then eventually cell suspension. Various biomass they are available, screening tools and AI they are available. We have to use them. Impact of the media on the cell flavor, that's another topic. The quality of the final products depends on the material that we feed them. Media sterilization without compromising the quality of the media. And the other thing is the spent media that we are generating through the cell culture. So in my program also we are addressing that as well. We are producing, for example, algae and single cell oil through circular economy process as well. Regulatory challenges, as you know, food safety and regulations, there is not that much information around this. People, they are working together. I am a part of different committees to improve that. I am also reviewing the food safety regulations related to the cellular agriculture for other countries. Like, for example, I do that for Japan as well. I have a project right now with the Japanese government. Policymakers and state regulatory agencies, they need to be involved. There's a lot of misinformation regarding all of the topics, not just this actually. And as you know, religious aspects of the cultivated meats also needs to be addressed. And there are 2 or 3 papers published in this area which are quite interesting and very informative. This year actually, so September 2024, we actually hosted a symposium and the third National Institute for Cellular Agriculture meeting here at Texas A&M, and we discussed the potential of the cellular agriculture as a revolutionary method for food biomanufacturing for space missions. And so right now we develop another proposal around that as well. It was a really interesting panel discussion, and we learned a lot actually by ourselves as well. And this is my team members on the top and our group from the National Institute for Cellular Agriculture. And I finished my presentation, and looking forward to discuss this topic more actually in the classroom with you. Go ahead and read about this. Go ahead and search and Google and read more about these topics. And I cannot wait to meet with you. Thank you so much.